Hi, my name is Prakash. I'm presenting a paper titled "A Universal Non-Parametric Approach for Improved Molecular Sequence Analysis" today. Uh, First of all, thank you so much for providing this opportunity in PAKDD 2024. Uh, I'm excited to learn about the ongoing research and share my work as well. So in this presentation, we will be discussing the introduction, the motivation behind the problem, the advantages we got from our proposed approach, the workflow, and the data set that we use to testify the results. We'll discuss the baseline models that we used, and then we finally conclude along with the results and discussions. So we have seen a widespread use of neural network in the molecular sequence analysis and classification problems. But these models often require a substantial number of parameters and a large data set to often work efficiently. On top of that, the high dimensionality of the nature of the biological data set makes it highly inefficient and computationally expensive. So there is a need of a low dimensional embedding for molecule sequence analysis, which can provide a better and efficient solution for sequence classification. So we came up with an idea where we can use data compression as a technique, which can help to provide a better representation when it comes to molecular sequence analysis. Traditional method for classification of molecular sequences includes feature-based embedding and kernel functions-based methods, the language models. We can use neural networks, but all these methods have certain limitations and demands huge data to work efficiently. Specifically, the neural network-based method requires substantial number of parameters and long training times, which makes it computationally expensive and resource demanding. Also, it requires a larger data set because we have to train the model, then only it will be efficient and avoid overfitting, which is a problem when it comes to biological data set because quite often time we don't have the huge data source and data sets. So these methods are definitely not a perfect solution when it comes to low resource or rare species scenarios for biological data sets. So we came up with a solution called, we call it the GZIP classification method. It is a novel approach to molecular sequence classification, which addresses the limitation present in the existential methods. Basically, it is have three main components. The proposed approach has three main components. The first one is we use the compression-based models, such as algorithm like GZIP and BZ2. Then we compute the distance matrix, which is a non-symmetric distance matrix. We use the normalized compression distance for that. And then we convert these distance matrix into a kernel matrix using a kernel PCA to get the low dimensional embedding. We'll discuss more in detail that how it functions, but like on broader terms, it employs the novel technique, which is based on lossless compressors and normalized compression distance matrix. So what was the motivation? As we all know that molecular sequences offers a profound insight for structural and functional and evolutionary characteristics. It plays an indispensable role in fields like drug discovery and evolution biology and gene annotation. And the availability of large-scale training data can be a significant bottleneck in developing machine learning models for molecular sequence analysis. As the number of parameters in neural network increases, the model's interpretability also often decreases, which is another motivation that why we want to work on this problem. So techniques like compression can be helpful in mitigating these challenges. So we tried to explore that how we can incorporate, and we came up with a proposed solution um, to tackle this problem. So what we achieved after proposing this approach and is the proposed method not only reduces the parameters requirement, but it is also lightweight and accessible, so it can efficiently handle the low resource biological data set as well, uh, which is very limited in biological context. The other benefits we have achieved are like we enhance the accuracy. The proposed method achieves superior accuracy as compared to the traditional methods and the baselines that we have compared. It gives you the improved efficiency since um, it can work for low resource data also, so it's more efficient in that way. And definitely it is more scalable because we are using low dimensional representation, making it more scalable. That more amount of data, if it comes, then we'll be able to handle it too. So coming to the methodology, we are using two algorithms like GZIP and BZ2 with NCD, the normalized compression distance, to compute the kernel matrix. So the first step involves is compress the sequence using either of these algorithm, the GZIP or BZ2. And then we compute the normalized compression distance between each pair of molecular sequence. And finally, we generate the kernel matrix using Gaussian kernel and distance matrix that we obtained from step 2. Finally, we employ the kernel principal component analysis to get the vector representation of corresponding molecular sequence. In this paper, we also provide the theoretical justification for kernel matrix from the NCD-based distance matrix. That's why we are using the NCD-based distance matrix. So as I've mentioned, the compression method that we are using are BZ2 compressor. It is a general purpose lossless compressor based on Burroughs-Wheeler transformation and Huffman coding. The second compressor we are using is a GZIP compressor. Uses very few bit to represent information, which is based on lossless compression algorithm, LZ77. It is a compression algorithm and a dynamic Huffman algorithm. The justification of employing the kernel matrix, why we using the NCD-based distance matrix, is because we get several benefits out of it, like the non-linearity, since it enables the capturing of integrate non-linear relationship. We justify the use of NCD-based matrix. It also captures the complex relationship. The Gaussian kernel captures the integrate relationship between the sequences. And we can capture the complex pattern, structure, and data, which are the issues in linear methods. So we surpass the limitation of linear methods in that way. We get the theoretical properties of Gaussian kernel because we are employing the Gaussian kernel. So we get benefited with the theoretical properties of the kernel as well. And the smoothness and continuity and sensitivity are also availed to enhance its ability to capture local relationship. We get the enhanced performance by capturing the complex relationship. The data enhances the classification of sequences, so we are getting the enhanced performance. This is the workflow. We have an input sequence S1 and S2. We concat these two sequences in B, and then we encode the sequences. The first step involves compressing. So we use the compressor to compress these sequences, and then we compute the length of sequence and find the distance matrix, the second big component. So we get the distance matrix based on NCD. And then we get the kernel matrix computation, the final step, and finally we employ the kernel PCA to get the vector out of it. So speaking of like evaluating our method, it's better than the deep neural network. as there is no need of pre-processing or training. So this all process makes it simpler. We need fewer parameters with no GPU resources are needed for distance matrix computation, making it lighter and absence of underlying assumptions. We don't have any assumptions about the data, then it makes it universal, as the title suggests for this paper. The algorithm is same as the workflow. Here we have the set of sequences as input, and the output is a distance matrix, which is NCD based distance matrix. That's what we propose in this paper. And after we get this distance based matrix, we get the vector out of it after applying the kernel PCA. So the data set that we used in this paper is the human DNA data set, which has 4,380 sequences, comprises of 7 different classes. These are unaligned nucleotide sequence to classify the gene family to which human belongs. So we have 7 gene family where the sequences of human DNA belonging to 7 different gene family, which is G protein coupled, tyrosine, and synthesis ion channel and transcriptor factor. These are the classes that we are trying to predict for the given input sequence. These are the statistics for the data set we are using. The sequence length is maximum is 18,921 and the minimum sequence length was 5. The average is around 1260. So it has a wide variety of unprocessed sequence data in here. That's what the ultimate notion of it. The baseline method that we use to compare our proposed approach are the feature engineering based method, the kernel matrix based method, which is string kernel, the neural network based like WDGR and autoencoder, we compare it with that too. The language model we use, the pre-trained language model called seqvec, and we use the pre-trained transformer to compare our result with protein BERT. So feature engineering based method, kernel based method, neural network based method, and language model and transformer. So we are pretty much comparing with all types of methods that could be potentially used in here. And the results we got are very amazing. You can see that the feature engineering based method, PWM2Y, we ran it on other feature engineering based methods as well, but PWM2VEC has been proven by authors as the best among the others, like Spike2VEC and Minimizer and Space Schemers. So we just representing one from feature engineering in the paper. So we can see that 31% accuracy. The kernel-based method, string kernel gave 73%. The neural network-based method gave us 69% with autoencoder. The WDGR was bad with 45% accuracy. The seqvec gave around 72%. Protein BERT performed 54% accuracy. Whereas when we run the experiment on the similar data set with our proposed method, you can see the significant difference. Like we got 83% accuracy when we compare from the previous one. The best one here is probably 73% with random forest and string kernel-based method. And here we got 83%. So it's like a significant 10% jump in providing the accuracy with our proposed method. And that applies on other metrics as well. So if you can see, like, it's almost 78, 74%. Here we got 85, 83, 83. So for all the metrics, you can see there is a significant difference in increasing the better performance. And the classification results here we are presenting are averaged over 5 runs for different evaluation metric. We can see it's very efficient as compared to other baseline methods. And in the main paper, we are comparing the results from standard deviation result and statistical significant results as well. And we can conclude that our proposed method outperforms the SOTA for predictive performance. To conclude, we have proposed a GZIP or compressor-based method. That is a lightweight and efficient compression-based model, presenting a promising new direction for molecular sequence analysis. It is simply combining the compression method with powerful nearest neighbor algorithm, and our method achieves better performance than the other state-of-the-art methods. We offer more accessible and computationally efficient solution, which will be very efficient, especially in low resource scenarios. And for the future work, we aim to um, experiment it for other bioinformatic domain, investigate further to optimize and tailor the approach for specific biological data set. So that's what we are looking forward to work on. I think that was it. And if you have any questions, I'll be happy to answer. Thank you.